And so I mentioned earlier in the lecture that oftentimes for these networks, you can see communities in the graphs. And so what do I mean by community? I mean, a community refers to a group of nodes for which there's a lot of interactions within the group of nodes, but relatively few interactions going outside of that group of nodes. And so communities within graphs oftentimes indicate a group of genes that have related function or somehow have strong relationships. So for example, in a protein-protein interaction network, a community will tend to point towards genes that form complexes, physical complexes, because in a physical complex you have a lot of connections between the different components of that complex, but relatively speaking, you have few connections going outside of the protein complex. Whereas in a genetic interaction network, for example, a community might represent an entire pathway, right? Because basically within a given pathway, you might expect there to be a lot of genetic interactions between pairs of genes in that pathway. In a co-expression network, a community would refer to a bunch of genes that are all co-expressed at the same time, and so they typically form. They also tend to point towards things like pathways, because basically different members of the same pathway might tend to be co-expressed, because if you need a pathway on, then all of the members of that pathway should tend to be expressed at the same time. If that pathway is not needed, then usually, many of those genes in that pathway will be shut off. And so genes within the same pathway tend to be co-expressed. Finally, in regulatory networks derived by, for example, ChIP-seq, modules in a graph will tend to correspond to genes that are co-regulated. So they're bound by the same set of transcription factors. So your module in a regulatory network might compose of both the transcription factors that regulate a target set of genes as well as the target genes themselves. And so a related term would be modular. And so a graph is said to be modular if it contains a lot of communities with very few connections between different communities. And so it's modular because there's clearly different components within the larger graph. And so an obvious question then would be, well, if I use, if I do a PPI assay or genetic interaction assay and I get a genome-wide interaction map, then how do I identify these different communities that might represent different groups of functional pathways and so on? And so there's many different pieces of software that are available for identifying communities in graphs. The list is way too long to list here, but what I will say here is the general principle upon which they work. And so the general idea about behind identifying communities in graphs is that essentially to take a single graph and separate it into subsets of nodes, where you say this bunch of nodes represent community 1 or group 1, this set of nodes represent group 2. Conceptually, what you're doing is you're taking a graph, and say, for example, in the case on the left here, where you have a graph with clearly two communities with a few edges between them. Conceptually, when you are trying to determine which genes belong to one community versus another, what you're conceptually doing is this is kind of like a two-step process, where in the first step you kind of propose a candidate division of the graph into two different communities, group 1 and group 2. So you predefine which nodes you think belong in group 1 and group 2. And then essentially these approaches basically define a score that say, okay, given a proposed grouping of the nodes of my graph into different communities or groups, how good of a division are these different groups? And so the way that groupings are typically scored by these methods is that when you take a graph and you separate them into two potential groups, for example, one way that scores are measured, one way that that particular division into communities is scored is by asking the question: if I separate my nodes into these two groups and I cut this graph, what is the minimum number of edges that I have to cut in order to completely separate the two defined groups? So on the left here, so the two graphs on this slide, on the left and the right, they actually represent exactly the same graph. So it is the same graph in the left versus right. The only difference is that I've proposed two different groupings of the nodes, right? So where each grouping is defined by either a blue circle or blue ellipse. And in both the left and the right case, I've divided the graph into two communities just to make this example easy to understand. And so on the left, if those are my two groups that I've defined, then in order to completely separate these two groups or communities, I would only have to cut or chop basically three edges in this graph in order to separate them. Whereas on the right, if those are my two groups that I've defined, then I would actually have to cut 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I would actually have to cut 11 edges in order to separate these two groups. And so intuitively, the separation of group 1 and 2 on the left is a good separation of nodes into communities because I didn't have to cut that many edges in order to separate those groups. Whereas on the right, it's a bad cut because I had to cut a lot of edges. And intuitively, just looking at the left versus the right, you can kind of see if the definition of community is a group of nodes that have a lot of connections within the group, but not a lot without or outside of the group, then the grouping on the left clearly fits that definition better than the grouping on the right. And so, the next obvious question then is, well, now that you've identified, say, communities or groups of nodes in the graph, then what do you do with that? So oftentimes, it depends on the question you're trying to ask. Oftentimes, the first thing you usually ask is, well, what do the groups of genes belonging to the same community do? And so to answer that question, then the most typical thing to do would be to go back to gene set enrichment analysis, actually, and go to a database like Go and just ask for each community individually, are there any known gene functions that are enriched in that group of genes, which then indicate what that group of genes do? Something a little more complicated that people oftentimes do also is they don't just build like a network for one type of cell and then just identify communities. What they do is they typically measure or they typically assay networks in different conditions. So for example, they might compare networks derived from normal hepatocytes versus like liver cancer, hepatocellular carcinomas. And they might compare, okay, which modules exist in the carcinomas that didn't exist in the normal hepatocytes, because that would tell you about which groups of genes are now starting to function together in carcinomas that didn't form a group in the normal cells. So the idea about detecting communities in graphs and therefore then using techniques like gene set enrichment analysis to characterize the function of communities within the graphs is a good idea in principle. I think in practice, it doesn't really work out that well because these graphs are extremely noisy, especially PPI networks, genetic interaction networks. They just have so many false positive and false negative edges that even if a group of genes truly form a community in the cell, it may not be evident from the assay that is the case because of these bad edges. And so in practice, community detection is not the only way in which you find groups of genes that the network implicates as being part of the same pathway. Another approach is through what's called guilt by association. And so the idea here is that two genes that are either directly connected by an edge in the network, or they're indirectly connected because, for example, they might share a lot of neighbors in the graph. So a neighbor of a node is just a node that is connected to that node. And so if two nodes, for example, aren't directly connected, but they're connected to each other through somebody else, then the idea of guilt by association is that you're likely to share the same function, right? So, and basically, the more connections gene A has with gene B, the more likely that gene A shares similar function as gene B. So that's what guilt by association means. And so how that's important and how that's used in terms of graphs is that suppose that you have performed a PPI, or suppose you've performed a genetic interaction assay, you've characterized your network, and you know, for example, that one gene in this graph is known to be in the DNA repair pathway. And so that node is colored in red in this graph below, for example. And so the idea of guilt by association is that using the graph, you can figure out which other nodes in the graph that node is well connected to, and those other nodes will tend to also be, for example, part of the DNA repair pathway. And so if you basically could imagine performing like a random walk, right, where suppose you started that single node that's colored red, and whenever you started that node, suppose you just pick an edge at random from that node and walk to that node that that edge implies. And you do, suppose you perform a lot of these random walks, what you would find is that the nodes that you visit the most frequently, so shown on the right, so nodes that are more red are visited more frequently, what you'd find is that the nodes that are visited most frequently are the ones that there are a lot of paths to that node from the starting red node. And so you can see from this random walk example that if I start a random walk within what looks like clearly a community, most of the nodes that I visit end up being nodes within that community, although sometimes I'll walk outside of that community, as indicated by the central node that has high betweenness, that has a little bit of red. And so the point I'm trying to make here is that in practice, because communities may not be obvious in your graph, sometimes guilt by association is used to say, okay, if I know there are some genes that perform, say, DNA repair, then I figure it out from Go, then I can use that to kind of take DNA repair labels, which I'm representing as red colored nodes here, and just do random walks to find other genes that are highly connected to those initial DNA repair genes in the network, to find additional genes that are likely to also kind of either participate in DNA repair or are participating in a related pathway. So I want to leave you with a few questions to think about. Number one of which is I want to circle back to this question of how many networks are there really in the genome? And so again, it's important to remember that interaction networks don't represent static things like DNA sequence. They're highly dynamic because interactions tend to depend on context, and context can change throughout different types of cells or different environments that an organism lives in. And so, an important question becomes, if your goal is to characterize the set of all interactions that happen in a genome across all possible contexts or cell types, is that an impossible problem? Like in humans, for example, where you have thousands of different types of cells and then, uncountable numbers of environments in which those cells can exist. Is this just a really hard problem and impossible to solve? Should we just give up and not try to comprehensively assay all possible interactions that can happen between genes in a given organism? Another thing to think about is that of time scale. And so if you think about interactions, say regulatory interactions in the context of perturbations, so what's common is, for example, to perform, say, ChIP-seq assays before and after perturbation. So suppose you want to understand how gene regulation changes in response to, say, chemical exposure. Oftentimes what you would do is you would take a collection of cells, you'd, say, run a couple of ChIP-seq assays before, sort of in the steady state, and then you would perturb the cells with your chemical exposure. and then try to perform your ChIP-seq assay again to measure what kind of regulatory interactions resulted from the chemical exposure. And so an important consideration is one of timescale, in the sense that not all responses to external stimuli happen immediately. It can take time for example, mRNA to turn over transcription factors to activate genes and start a cascade of regulatory events that ultimately lead to some kind of response to that stimulus. And so if your goal is to perform ChIP-seq assays after perturbation, you have to think about the time scales of how long does it take for the cell to respond, because if you run your ChIP-seq assay too soon after exposure, you might not capture all the regulatory interactions that occur as a result of exposure. A related question is, what type of network should I collect? Right. So if your goal again is to understand how carcinomas develop, it may not be sufficient to just collect a lot of co-expression networks just because RNA sequencing is easy and cheap to do. There's a lot of metabolic interactions or protein-protein interactions that appear or disappear as carcinomas develop. And so oftentimes you need to think about, how do I, of all the possible assays that I could do, which ones are most important to the interactions that I'm trying to find. And the final question I want to leave you with is the question of conservation of interactions. So I briefly mentioned that genetic interactions seem to be poorly conserved, at least between different yeast species. And so again, one of the primary reasons why model organisms, for example, are model organisms, is because people hope that the interactions and the regulatory behaviors at the molecular level anyways, that you can learn from model organisms like mouse might also exist in humans as well. And so an important question then there is, which interactions do you think are most likely to be conserved between, say, model organisms and humans? For example, you might expect that interactions that occur that help maintain the proper cell cycle process are more likely to be conserved than, say, processes associated with neuron development, for example. And so it's just important to think about, if I'm using, if I'm performing a set of assays in model organisms as a model for humans, for example, which interactions are more likely to be conserved and therefore useful to study in model organisms for the purpose of informing us about humans.